Alright guys, so we just left off talking about RNA processing or pre-mRNA processing. Now let's talk about RNA splicing. Okay, so let's say we have our mRNA strand here, right? Our pre-mRNA strand here. We already added the 5' cap, we already added the 3' poly A tail. We still have to splice out the intron though, because remember, the exons are the only parts of this RNA strand that's going to go on to form or go on to be translated into proteins, right? Introns are not, so we got to splice these out. Okay, so how are we going to do that? So let's take this scenario, right? We have our exon A, right here, from the green line to the orange line. Then we have our intron from the orange to orange, and we have our exon B from the orange to yellow. Okay, and then this is our whole 5' to 3' RNA strand. Okay, so what happens is in the middle of this intron, we're gonna have an adenine. Attached to it, we'll have an OH group. Okay, then what we're gonna have is we're gonna have a GU here and an AG here. Okay, so we're gonna have a GU guanine uracil here and an adenine guanine here on the sides of the adenine. Okay, so remember this side, right? This side of the guanine is the 5' end. Okay, because we're over here on this side. I'm saying this is the 5' end. So on this side is the 5' end. Remember, our bases, right? We have our bases, then we have our sugar phosphate backbone, which is connected through phosphodiester bonds, right? So we have our sugar phosphate backbone, which is held together through phosphodiester bonds. So the 5' end, remember, is the phosphate end. Okay, so that's why we have a phosphate shown here, which is connected to the oxygen, right, on the alcohol part, right, of the 3' alcohol of the sugar. So the 5' phosphate is connected to the 3' alcohol or the 3' oxygen, right, of the next nucleotide. So again, right, we have our GU here. Okay, I know it sounds a little confusing, but just remember the basic structure of our sugar phosphate backbone. We have our 5' phosphate end, which is right here, right? And then that connects to the 3' oxygen, okay? The 3' oxygen. And then that oxygen is connected to of course, the sugar, right? The ribose sugar. And then again, it repeats. So remember, I can pull up a picture real quick. All right, so here's a drawing that I quickly drew right on the side just to explain this better. So remember, this right here, okay, on this side is the 5' side, right, for the guanine. So like this is the 5' phosphate. So what I mean by that is right here, right? Let's say we're talking about this position right here. This is the 5' phosphate because it's 1, 2, 3, third carbon, fourth carbon. Sorry, the fourth carbon's right here, right? This is the fourth carbon. Then this right here is the 5th carbon, right? Off the 5th carbon, we have the phosphate. So that's this phosphate right here, right? That's the 5' phosphate because it's on the 5' side, right? It's on this side. Then this 5' phosphate is connected to this oxygen, right? On this sugar. So on this oxygen, what's the carbon number for that oxygen, right? 1, 2, 3. So that means this is the third, the oxygen that's connected to the third carbon of the sugar, right? Of the next sugar, which is this one. Okay, so and then right, this oxygen, see how I didn't draw it in more, because this oxygen will then be connected to another nucleotide, right, which in this case is right here, right, because on the 1 carbon is going to be another base, right? So if we're talking about guanine, right, you know how we were talking about guanine right here, so that's what we would have here. We would have literally guanine off this, right, guanine, and then this would this would be the 5' phosphate, which we're talking about right here, right? And then this 5' phosphate is connected to this oxygen, which is the 3' oxygen of the next sugar, right? The next nucleotide, or the next sugar of that nucleotide, right? Which is right here. So what happens is the O that's part of the OH of this adenine will attack the phosphate, okay? Will attack the phosphate. So let's erase all this here, right? What's going to happen is the O, right, this O will attack the phosphate, okay, will attack this phosphate. And then that will cause this bond right here, this phosphodiester bond that we just showed, to break. So then what's that going to lead to? That's going to look like something like this, right? See, this oxygen now will gain these electrons and then a hydrogen will bind to it. So we're going to have an OH now, a hydroxyl sticking out of this exon A. And then remember, this O attacked the P, so we formed a bond between the O and the P. So where can we locate that? Well, remember, this A is this A. Again, this A is this A. This adenine had the O on it, right? OH attached to it. So that's the O, that's the O. This O right here attacked the phosphate. So that's this bond right here, right? Then this phosphate, remember, was bound to the GU. So it's still bound to the GU. So we created this weird loop, right? Next, what happens is this hydroxyl that we just created, right? This oxygen will go ahead and attack this phosphate, okay? And then this bond will break, okay? And then we'll have a formation of a bond between this oxygen and this phosphate. So we're going to end up having oxygen, this oxygen, phosphate, this phosphate, oxygen, this oxygen, and then bound to the rest of the nucleotides. Okay, so we formed a new bond, right, between this oxygen and this phosphate. All right, and see, now we have two exons linked together, right, through phosphodiester bonds. So we basically snipped out the intron and joined the two exons together. That's how splicing works, okay? But we have to just learn one more thing. Now, this last thing we have to focus on is we can't really do any of this splicing that we just went over without the use of SNRPs. Okay, these right here, SNRPs, small nuclear ribonucleoproteins, right? Well, how do we make these SNRPs? Remember RNA polymerase 2? It also synthesized small nuclear RNAs. We can take these small nuclear RNAs and combine them with combine them with a specific type of protein called Smith proteins. If you combine small nuclear RNA with Smith proteins, you get small nuclear ribonucleoproteins, or SNRPs. These SNRPs are really important in this splicing process, okay? There's two main SNRPs that we should be familiar with. One is called a U1 SNRP, and another one is called a U2 SNRP, okay? The U2 SNRP, right? The U2 SNRP can bind around right here where the adenine is, right? So it's going to bind right here near the branch site, or the branch site, right? The U1 SNRP can bind around right here, right? Okay, so the U1 SNRP can bind around right here. So without these proteins, it's going to be very hard to do the splicing process. Okay, so remember U1 SNRPs and remember U2 SNRPs. Remember Smith proteins also, okay? Because in the video after this, we're going to go over the clinical relevance to all this. All right, so see you guys there.